which any researcher interested in a particular cellular process can use to answer three key questions. First, what are the next data that I should collect to understand this process better? Second, how useful are those new data once I've collected them? And third, what are the new biology that actually came out of those new data? Now, the heart of this method is a certain type of model which we call a data-driven gene ontology. Now, what is that? Here's an up-close look at one of these. They're hierarchical cellular models where each of the genes, represented as ovals here, are annotated to terms, represented as rectangles. And each of these rectangles is a function that the cell has to perform. It's a hierarchical model because these functions are nested within each other, so that the most specific functions are at the bottom and the most general are at the top. Now, the format of this is highly similar to the manually curated gene ontology, which many of you are likely familiar with. So why is this called data-driven? Well, because it's inferred automatically and computationally directly from network data. And by doing this, we are extracting useful information and displaying it in a way that both humans and machines can understand with these data-driven ontologies. We like to think of this as analogous to structural biology, where you extract useful information in the form of crystal structure from an initially difficult to interpret X-ray diffraction pattern. Furthermore, we have some previous work which showed that these two structures, the networks and these data-driven gene ontologies, are actually informationally equivalent. So we can interconvert freely between them. And we're going to use that fact today for active interaction mapping. Okay, so back to AIM. This is the meat of this presentation, and we want to answer these three questions here. How do we go about doing that? So we start off by gathering up all of the publicly available network data that we can get our hands on. And then step one is to integrate all of that data into a data-derived gene similarity network. And we do this by turning this into a supervised learning problem. So we start out with all of this network data, which we can of course just represent as a table where each of the rows are gene pairs, each of the columns is a different type of experimental data, or actually one experiment. And then we can take known biology in the form of the gene ontology, and we can use our previous fact and just convert that into a network. And that turns this into a supervised learning problem where we can just do classic machine learning, learn patterns on part of the data, and predict on the other part until we've got our integrated network combined from this experimental data. So we've learned what experimental data supports a strong, close relationship between any pair of genes. Once we've done that, a measure of performance can be just a simple Pearson correlation between our network and the Go semantic similarity network. Okay, so back to AIM. The next step after we've created this combined network is to convert that network to a hierarchical model. And that method was already previously published, but just to give you some intuition, here's kind of an idealized case where on the left we have an idealized network, and on the right is the ontology that we inferred from that. And basically what we do is we look for these nested groups of genes, and each rectangle you see in this network here turns out to be a term in this ontology over here. Now this is an idealized case. Of course, the real data looks a little bit messier, and if you're interested in hearing how we deal with that, please check out the paper reference down below there. So at this point, I'm going to switch from the general to the specific and just show you how we actually use this in our lab to learn about some new biology. And we decided to look at the process of autophagy, but everything I'm going to show you could easily be applied to any other cellular process that you're interested in. Just a little background on autophagy. It's a cellular system for nutrient recycling. So if the cell is either starving or has an overabundance of a particular organelle, it can scoop those things up, send them to the vacuole, and break them down to basic building blocks that can be reused. It's a highly conserved process from yeast all the way up through humans, And dysfunction in autophagy has been related to both to a wide range of diseases from cancer to neurodegeneration. So we're going to use AIM to study autophagy. How do we make it specific to autophagy? Well, we start with a core set of known autophagy genes, and then we look at a network similarity measure, and we just grab a whole set of genes which show similarity to our core autophagy genes in the network. And we include those genes in the model. It's about 472 genes. So once we've done that, we've run through this pipeline all the way to building a first-generation hierarchical model of autophagy, which we call ATGO, which is short for the autophagy ontology. And now we're ready to start answering these questions I promised you at the beginning. So step one, what are the next data that we should collect? To answer that, we want to try to find out what were the most important data so far for building this model. So we start dropping types of data from the model and watch how our performance degrades. When we do that, we see that the most important data were protein-protein interactions and genetic interactions. And then remember, the performance measure here is a Pearson correlation between our integrated network and the Go semantic similarity. So that's nice to know what was valuable so far, but where do we go from here? To answer the question, what data are most likely to improve the model, we drop individual studies. So not all of the protein-protein interaction data, but just one lab's experiment, usually a high-throughput experiment, and watch how the model degrades. And so when we do that, we see that the steepest slope, the quickest degradation, comes from the genetic interaction data. And so based on that, we decide that's probably the right data to collect to make this model even better. So now we know the data to collect is genetic interactions, and we went ahead and did that. We collected some new autophagy-specific genetic interaction data. So we actually have an advantage over previous experiments because we're not collecting just in a rich media, sort of non-specific way, but we can also collect data under autophagy-inducing conditions with rapamycin and with starvation. And we can look at how the genetic interactions change across these conditions. So now that we've got that data for about 156,000 gene pairs across all these conditions, we want to answer the question, how useful are those new data? And we can do that in a global kind of way by just throwing that new data into the mix here and watching how the model performs. And when we do that, we see a significant bump in model performance by including this screen in addition to all prior data. And in fact, this targeted screen designed to look at yeast autophagy actually contained more information than all the prior omics data that we could find. So now that we've got a measure of how useful are these data globally, we want to answer what exactly are the new biology in this data. And there's a couple ways to do that. The first way is to just look at how the model changed when we added that new data. Each of those changes we can attribute directly to information contained in that new data. And we have computational tools that help people do that. You can align the two ontologies and pinpoint exactly where those differences lie. But on top of that, we discovered that these models are very intuitive for biologists to look at. So if we just start showing one to a biologist, they immediately start coming up with testable hypotheses and ideas for what these new connections might represent. So we went ahead and did that. We showed our autophagy ontology to some expert biologists at our university. And each of these little Roman numerals here represents a bit of biology that we followed up on in the paper. But I'm just going to talk about this Roman numeral number 1 at the top here for today. And this was a term which in the original ATGO 1.0 contained just ATG 11 and 19. That's on the left side here. But including our new data, 2 new genes, ATG 26 and 27, were brought in. And when we showed this to our experts, they were kind of surprised because there was some previous very thorough work testing all the standard autophagy assays that showed ATG 26 was not involved in Saccharomyces, at least in autophagy. But given the placement of it in this ontology, they kind of started thinking and came up with a more nuanced experiment that might show ATG 26 actually is involved with this PR81 aggregate processing, which is kind of the core function of ATG 19 and 11. And so we did those experiments, and we did in fact find that ATG 26 do show a defect under these particular conditions, both quantitatively and qualitatively on microscopy. And we have a few other results like that in the paper you can check out below there. So I hope I've convinced you today that active interaction mapping is a useful new tool that you might be interested in using yourself. If so, please check out our website at acgo.ucsd.edu. You can run active interaction mapping yourself. You can explore the current ACGO model. You can download the Jupyter notebooks. They're available in both yeast and human. And if you have an idea for how to improve any step of this method, it's as easy as just changing a line of code in those notebooks. So I just want to thank all the co-authors on this paper that worked very hard, and my advisor, Trey Eideker, and all you guys for listening today. Thanks. Yeah.
Can you predict how much knowledge we could get based on these analyses? What's that for?
Yeah, so the question is, can I predict how much knowledge we could get from these technologies? I think if you just kind of keep doing similar experiments to what's already been done, then you can look at like the asymptote on one of them, like the plot that I showed before. But I think what we saw, with our new data is that if there's something qualitatively different about the type of data that you're collecting, then it's hard to predict based on past data how much information you'd get from that. Yeah, sure. So the question was, how do we turn this into a supervised learning problem? So we start with, we use each of the networks then as a feature in each of the rows in that table was a gene pair. And then the thing we're trying to predict is a semantic similarity value, which we've calculated from Go. So that's also just for each gene pair, we have that semantic similarity value. And then we use random forests to try to learn what experimental data supports a strong relationship in Go. So if two genes are known to perform some very specific function, then we found usually they have a lot of edges in the networks supporting them. And so we can learn new biological relationships by finding things that have lots of experimental data behind them, but maybe aren't yet in Go. Okay. Thanks.